Assalamu alaikum. First of all, I would like to thank the organizers of Third International Brain and Biomedicine Conference for giving me the opportunity. My name is Syeda Zahra Batool. I am Associate Lecturer in Women University of Azad Jammu and Kashmir with MPhil in Microbiology from Abbasian University, Islamabad. Today, I am honored to present my research on the topic isolation and identification of cholesterol-lowering bacterial strain from indigenous Cucumis sativus in Pakistan. As we all know, probiotics are living non-pathogenic microorganisms. When consumed in sufficient amounts, they provide health benefits to the host. Lactic acid bacteria have probiotic properties. One key benefit is reduction of serum cholesterol. The major probiotic genera are Bifidobacterium species, Lactococcus species, Lactobacillus species, and Streptococcus species. The main objectives of my study were to isolate beneficial probiotic bacterial strains from Cucumis sativus, to determine cholesterol-lowering ability of probiotic bacterial strains, identification and molecular characterization of cholesterol-lowering probiotic bacterial strains. It is studied that pharmaceutically available cholesterol-lowering drugs have some adverse side effects on humans. So this study provides knowledge about naturally occurring cholesterol-lowering bacteria in Cucumis sativus. First of all, 30 samples were collected from different areas of Rawalpindi and Islamabad. Samples include fresh Cucumis sativus transported to lab in sterile containers and stored in refrigerator. Isolates were diluted tenfold using serial dilution in PPS solution, inoculated on MRS agar plates and incubated 37 degrees centigrade for 24 hours. Organisms identified on the basis of morphological and biochemical tests. This all study was conducted at NIGAP, National Institute of Genomics and Advanced Biotechnology, NARC, Islamabad. These are the pictorial representation of colonies on MRS agar plate and different biochemical tests performed on the isolates. This is the tabular representation of colony morphology of isolated strands. This is the pictorial representation of bacterial colonies on MRS agar plate. And from total 10 isolates were catalyst negative, so they do not produce bubble and remain settled down in hydrogen peroxide. Only 2 isolates were catalyst positive as they produced bubble in the solution. Then we performed gram staining of all the 8 isolates. Only 7 isolates were gram positive as they appear purple bluish in microscope, while one isolate was gram negative as it shows pink reddish color in microscope. Then we performed different enzymatic potential tests. All 7 isolates show positive results with amylolytic and proteolytic test. And for DNase test, out of 7 isolates, only 6 isolates were DNase negative as ozone inhibition found around Strickland, while one isolate showed positive results. Then we perform antibiotic sensitivity test. This is the pictorial representation of antibiotic sensitivity test, and this is the tabular representation of antibiotic sensitivity test. 6 isolates show susceptible results against following antibiotics like vancomycin, metronidazole, tetracycline, penicillin, amoxicillin, erythromycin, gentamicin, ceforaxime, and bacteriocin, while resistant and intermediate against the rest of the antibiotics. Then we grow our colonies at different pH levels. This is the pictorial representation of colony growth at different pH. And all 6 isolates show better survival rates at bile salt concentration at 0.5% and 0.3% bile salt concentration. And this is the tabular representation of cell viability. Then we performed PCR of all the 6 isolates, and only 4 isolates showed DNA band in gel electrophoresis, which were sent for sequencing later in order to determine nucleotide sequence of DNA bands. We use 1 kb ladder, then we run control, negative control, and all the 4 samples that show bands on the PCR bands on gel document. And all the positive PCR positive samples then sent for 16S rRNA gene sequencing to Microgen Korea for sequencing using 16S universal primers. Partial Sanger sequencing was done. Out of 4 isolates, only 2 isolates, that are Z into 10 raised to power minus 4 and Z into 10 raised to power minus 5 nucleotide gene sequence was obtained. Sequence of Z into 10 raised to power minus 4 consisted of 999 nucleotide base pairs, and nucleotide sequence of Z into 10 raised to power minus 5 was comprised of 1372 nucleotide base pairs. After the BLAST results, multiple sequence alignment is performed through MSA tool of NCBI database to check the similarity of 10 Z into 10 raised to power minus 4 and Z into 10 raised to power minus 5 sequence with already present sequences of Pediococcus pentosecius and Lactococcus lactis in database. Then we perform the isolated and Z into 10 raised to power minus 4 was isolated as Pediococcus pentosecius sequence with a similarity of 99.70%, while Z into 10 raised to power minus 5 isolate was identified as Lactococcus lactis strain with sequence similarity of 96.97% as shown below. After multiple sequence alignment, phylogenetic analysis was performed by MegaX tool to study the evolutionary relationship of isolated strains, and this is the phylogenetic tree of Pediococcus pentosecius, and this is the phylogenetic tree of Lactococcus lactis. Then we perform cholesterol lowering activity assay. Cholesterol absorption by probiotic strains were determined by inoculating the bacterial strain at 37 degrees centigrade in MRS broth having 0.30 ox-gal. Water-soluble cholesterol was filtered, sterilized, and then added to the broth at a final concentration of 50 to 200 microgram per milliliter. Average strain was inoculated at 1% level and was incubated at 37 degrees centigrade for 24 hours in anaerobic conditions. After incubation, mixture was centrifuged 10,000 gram 4 degrees centigrade and for 10 minutes. Then supernatant was collected. Modified colorimetric method was applied to analyze the concentration of cholesterol in the supernatant. This graphical representation of cholesterol lowering activity assay of isolated probiotic strains result shows that both the tested strains had increased cholesterol reducing level ranging from 35 to 45 gram per milliliter, whereas the cholesterol elimination fell to 15 to 20 gram per milliliter in medium lacked bile salts. Compared to the other tested strain, Lactococcus lactis absorbed more cholesterol from the growing medium. I finally conclude my study that the most probable probiotic options among some of the studied isolated bacteria were Pediococcus pentosecius and Lactococcus lactis, both of which possessed exceptional functional qualities, specifically cholesterol lowering properties. Future research involving animal testing of such strains is required to confirm the probiotic properties, mechanism, safety, and application as a food additive or in conjugation with a biotherapeutic. Thank you so much.